Good afternoon. Welcome to our next forensic toxicology lab. So today we are doing forensic chemistry drug detection. So in the last lab, we observed testing for cyanide, which was a toxin or a poison that we found located in some medicines that are commonly found at the home. Today we actually have a little bit different of a scenario. So I'm actually going to read the scenario to you and then we're going to start the lab. One day at school, shortly after lunch, a large number of students approached the school nurse complaining of stomach pains and nausea. Never had so many students approach the nurse in one day, so school officials asked the ill students a series of questions. After several questions to each student, it was discovered that one thing that they all had in common was that they had had the chili offered in the cafeteria for lunch. Nervous about the possible causes, school officials immediately called the police. When police investigators arrived and confirmed the students' stories, they immediately ordered every science teacher to take inventory of all chemicals in his or her lab, as well as asking the school nurse to inventory all medicines in the school infirmary. Science teachers reported back that everything was accounted for. However, the school nurse reported that a large bottle of aspirin crystals was missing. She typically kept them on hand for students that didn't like to take pills, and she could mix it in with different types of juice. Alarmed, investigators approached the cafeteria staff and asked what ingredients they used in the chili, particularly anything white and either crystal or powder-like that could resemble aspirin crystals. Cafeteria staff told investigators that beside the basic ingredients and spices that did not match the description, they do add salt for flavor, sugar to reduce acidity, and cornstarch as a thickening agent. Investigators have confiscated the sugar, cornstarch, and salt, and now they have been sent to you to test to see which one of them could have been tainted with the aspirin crystals, if any at all. So what we have with us today, we've got a few different things. The first thing we have is the actual control. So this is aspirin crystals. Then these are the bottles that were confiscated from or samples of the items confiscated from the cafeteria. So we have sugar, we have salt, and we have cornstarch. We also have some chemicals here. We have Lugol's iodine. We also have a ferric nitrate. We also have sodium hydroxide. Those we're actually going to be using today to determine which one controls the aspirin. The reason why that is so important is because if the aspirin is present, when certain chemicals react with it, it will turn the color that we're looking for. So a few things you need to know. Acetyl salicylic acid, which is what is contained in aspirin, decomposes. When it is actually exposed to ferric nitrate, it forms a red-brown color. However, if it is present in another compound, it will turn a deep purple color. Now, the other thing we have to pay attention to is that in the presence of starch, brown iodine will turn into a blue-black substance. We saw that when we did the starch iodine test for saliva on the samples of cloth during the presumptive lab that we did to test for blood and saliva. So here we go. The first step is we're going to fill each one of the wells with samples of the control, the sugar, the salt, and the cornstarch. I'm obtaining a lab scoop here, and I also have, I'm going to grab some paper towels. I don't want to mix the substances together on the lab scoops. Okay, so the first one I'm going to do is I'm going to fill each one of my wells with a sample of the control. doing my best not to spill it into the other wells. We don't want any cross-contamination of our substances. Okay. The next one I'm going to fill is the sugar. We have that labeled SU up here on top of our plate, and I'm not sure if you can see that, but our plate is labeled. And these granules like to go all over the place. Spilled some of that sugar out. I really just need enough in there to have a decent sample to test. Okay, next one's going to be the salt. And in just a minute, when I get done filling the plates, I will give you a better look at all of this. And then the final one we're going to test is the cornstarch. Okay. I also needed some distilled water, so I have that as well. And now we can actually begin taking a look to see what type of reactions we are going to have with the different chemicals. So I am actually moving the camera so you guys can see this a lot better. I apologize, trying to get this in a good spot for you so you can see the reactions. And the camera just fell. No one should hire me as a video person, ever. Okay, so there's a few things that they're asking us for in our data analysis. So this is going to be a qualitative analysis, and so it is asking us for physical appearance, and then it's asking us for what things look like once we start adding the sodium hydroxide, the Lugol's iodine, the distilled water, etc., to see what colors we get or what type of reaction we have. So on the first one, I'm going to move this over a little bit. I'm trying to get the light better. It's reflecting here. Hopefully that helped a little bit. Dim the lights. Okay. So the first thing it wants us to do is add a couple of drops of water across our control, our sugar, our salt, and our cornstarch. But before we do that, we want to annotate what the physical appearance is. So the control to me is very granular, white. Same thing with the sugar, same thing with the salt. And then finally the cornstarch is more like a white powder. Okay, now I'm going to add the water. And again, it's asking us distilled water, of course, because it contains no chlorine or chemical additives. So I want to add enough water in here that I can get a good analysis. See what happens here. Okay, I have some toothpicks here, so I'm just going to use a different toothpick, of course, for each mix here. So this is kind of turning a frothy, kind of cloudy color. It's not real clear, kind of clumpy. And then with the sugar, it's definitely almost clear. It seems like it's blending quite well. Still staying pretty granular. And then the salt really seems to kind of just be melting in the water. There really isn't any grain left. It is still cloudy though. And then let's see what happens with the cornstarch with the water. It's real milky. It almost looks like milk. So I'm going to document my qualitative analysis really quick. With the water in the control, it's still pretty granular, cloudy. With the sugar, it's granular, kind of clear. I'll give you a close-up of this in a second. And with the salt, it's cloudy, but it's mixed. There isn't really a lot of granularity there. And with the cornstarch, it's very milky. So let's see how I can get you guys to see close up what we have here. So there's the corn, that's the control with the water. There's the sugar with the water. There's the salt. And there's the cornstarch. All right, so now, next steps. Is to add the iron to the mix. Okay, so the iron is our ferrous nitrate. So we're going to be combining the sodium hydroxide and the ferrous nitrate. And so we're going to add a couple of drops of the sodium hydroxide to the samples in the row labeled iron. We're going to wait a few seconds, and then we're going to add the ferric nitrate in the row labeled iron. And then we're going to record our results in our data sheet. So step number 1 is the sodium hydroxide. One, two, three. 1, 2, 3. 1, 2, 3. 1, 2, 3. All right, that's the sodium hydroxide. Then it says to add a couple of drops of the ferric nitrate. All right, and see those color changes. And then we want to document those color changes. So the first one turned a really dark, the control turned a really dark reddish brown. A little orange in there. Kind of an orangish purple, actually. Looks like the sugar is the same color. The salt actually just looks like a real rust, like a rust color, a yellow brown. And the same with the cornstarch. Sort of a rust color. Let me see if I can get it close so that you guys can see what those look like. All right, now the last step is going to be adding the Lugol's iodine to the mix. So the final step is a couple of drops of the Lugol's iodine in the row labeled iodine. So let's see what happens with Lugol's iodine here. Look at that turning almost black. All right, when we mix the iodine in with the control, again, it's this rust color. And then down here at the bottom, this is, remember, iodine turns this black, blue-black color in the presence of starch. So we know the cornstarch is reacting the way it should. So now let's look at our samples overall and see what we can see. So here's the control mixed with the sodium hydroxide and ferric nitrate solution. And if you look at these two, they are almost identical in color. And that's kind of the reaction that we were looking for. So based on this reaction that we have with those two being identical in color and having the exact same reaction, we can actually conclude that the sugar actually contains some of the aspirin. So I hope you enjoyed watching the color changes on this lab. It's one of the more fun ones that we do. And now we can work on the assignment. Thank you for joining.